Hello everybody, my name is Lauren Melendez and I'm a rising junior at the University of Pennsylvania. This summer I had the opportunity to work alongside the Cancer, Immunity, Infection, and Education Research Team, or Canine Research Team, spearheaded by Dr. Jennifer Punt. The group's objective was to better understand canine immunity in an effort to advance both canine and human medicine, as dogs are phylogenetically more closely related to humans than traditional model organisms used in science. What first piqued our interest were myeloid-derived suppressor cells, or MDSCs. MDSCs are immune cells that quiet the immune response after a pathogen invades and incites inflammation. However, little is known about how to distinguish them from regular inflammatory immune cells. Thus, it became our goal to look into distinguishing the classical monocyte from its anti-inflammatory counterpart. I, along with my partners Emma Hunter and Claire Dignazio, decided to look into a growth factor known as IGF-1 to see whether its receptor played an inflammatory or an anti-inflammatory role during an immune response. We found that the expression of its receptor, IGF-1R, was correlated with an inflammatory phenotype in canine monocytes. Two different types of stains were conducted using whole blood from 16 dogs, and two different methods were used to look at IGF-1 in the blood, which I'll go into in detail now. As shown in the leftmost diagram, IGF-1 is capable of binding its own receptor, IGF-1R, as well as insulin receptor. This middle diagram shows the two different methods that were used to locate IGF-1 receptor in the blood. This one involves a fluorescently labeled antibody for IGF-1, which is more typical of flow cytometry. However, we feared that this might bind to Fc receptors. This top diagram shows another method that we used to take a more physiological approach. We decided to use fluorescently labeled IGF-1 itself, eliminating the need for an antibody, but we thought that this might bind both receptors and cause extra fluorescence in the flow cytometer. However, both methods proved to be useful, and this gave us more confidence in our results. Our main goal was to determine whether IGF-1 receptor played an inflammatory or an anti-inflammatory role during an immune response. In other words, would its expression be increased on classical inflammatory monocytes or on anti-inflammatory suppressive monocytes? To test this further, we wanted to measure the expression of another inflammatory marker. We chose CD11b, which is an integrin protein and a marker of activation status for myeloid cells, as activation status and inflammation are often associated. To identify monocytes in peripheral blood, we gated out red blood cells first, and then we gated for CD14 positive cells, as the protein CD14, a co-receptor for TLRs, is widely regarded as a marker for monocytes. Then, to distinguish anti-inflammatory monocytes from inflammatory monocytes, we used a gating scheme designed by Sampath Kumar and Alex Cream. The CD14 positive population is thought to contain both MDSCs and intermediate monocytes, both of which are anti-inflammatory. The CD4 negative population is thought to be comprised solely of classical inflammatory monocytes. Gating scheme served as a basis for our analysis. The data shows that CD14 expression increases as IGF-1R levels increase. This suggests that the percentage of monocytes increases as IGF-1R expression increases. We began by gating low, intermediate, and high levels of IGF-1 receptor and then looked at the CD14 expression of each of those populations. This was done for all dogs tested using the two different methods of looking for IGF-1 that were described previously. The pattern occurred in all 12 dogs tested, using both of the aforementioned methods of looking at IGF-1R. The graph shows CD14 expression at each level of IGF-1 receptor expression, low, intermediate, and high, for all of the dogs tested. The left side shows the CD14 levels of those dogs whose samples were prepared with fluorescently labeled recombinant IGF-1, while the right-hand side of the graph shows the CD14 expression of those dogs whose samples were prepared with fluorescently labeled antibody for IGF-1 receptor. And although the levels of fluorescence differ among methods, you can see that the same pattern holds true. That is, that the percentage of CD14 increases as IGF-1 receptor increases. The previous figure suggested that monocytes upregulate IGF-1R, but we then asked which population of monocytes is doing this. Using our sample stained for CD4, we gated for monocytes and distinguished between classical inflammatory and non-classical anti-inflammatory monocytes, as shown here. We found that there was a higher expression of IGF-1 receptor found in the inflammatory subpopulation of canine monocytes, as pictured in these histograms. Interestingly, the expression of what we believe to be classical monocytes was increased further in sick dogs than in healthy dogs. For example, this top graph pertains to a healthy dog, whereas the bottom one pertains to a dog with a lymphoma, and as you can see, there is a stark difference, and we believe that this is something worth pursuing. All of this suggests that IGF-1 receptor is correlated with an inflammatory phenotype in canine monocytes, but we wanted to confirm this with another inflammatory marker, CD11b. We again gated for monocytes and then low, intermediate, and high levels of IGF-1 receptor and found the same pattern we did for CD14. That is, that CD11B is upregulated in canine monocytes as IGF-1 receptor expression increases. This was true for all of the dogs tested. And a graph of the MFI, mean fluorescence intensity, confirms this. This concludes my presentation. Thank you all so much for watching.